Imagine being handed a massive construction project. The absolute minimum time required to finish the work is 40 minutes, but your unchangeable deadline, 20 minutes. For a simple bacteria cell, this is the daily reality of reproduction. It takes 40 minutes to copy its entire internal blueprint, yet the cell itself splits in two every 20 minutes. That blueprint is DNA, and you can't just photocopy it. To replicate this data, the cell has to physically pull the entire twisted structure apart and rebuild each half, piece by piece. If the cell rushes this process or skips a single step, the genetic data is lost. To survive, it relies on a specialized workforce that manages multiple production lines simultaneously. Solving this 20-minute paradox is a matter of pure logistics. By organizing workers into overlapping shifts, the cell ensures the work is finished before the deadline hits. This replication project always begins at one specific designated location on the circular DNA loop, known as the origin. This diagram shows the first worker on the job, the unzipper. Its role is to act like a physical wedge, forcing the tightly wound strands of DNA apart to expose the raw code hidden inside. But here is the first bottleneck. The main builder machines that actually copy the DNA cannot start a job on their own. If they try to grab onto these bare, unzipped strands, they just bounce off. To solve this, a prep team drops a tiny chemical sticky note onto the strand. This note acts as a temporary marker that gives the builders a place to hold on. Now the builder machine locks onto that marker and begins racing down the line, assembling the missing half of the blueprint as it goes. This process happens in both directions at once, moving away from the origin to copy the circular loop from two sides at the same time. By dividing the labor into three specialized steps, unzipping, marking, and building, the cell creates a predictable, repeating workflow that can handle massive amounts of data. Even with these two teams working at full speed, the job still takes 40 minutes to complete. The cell is still dividing every 20 minutes. The cell solution is to start the next project before the first one is finished. It uses overlapping shifts to get ahead of the clock. This diagram shows the logic in action. Here, you can see a second round of replication beginning on these loops before the original blueprint has even finished separating. By starting the next round early, the cell ensures that when it finally divides, the daughter cells are already born with their own blueprints partially copied and ready for the next cycle. Biologists refer to this strategy as multifork replication. Because the cell is always working 1 or 2 cycles ahead, it hits its 20-minute deadline by distributing a 40-minute job across multiple starting points. But starting new shifts so early creates a risk. How do the workers know which parts of the blueprint are already done and which ones need to be copied? The cell uses a system of chemical tags. As soon as a section of DNA is copied, it receives a distinct chemical mark. A specialized bouncer protein reads these marks and physically blocks new workers from entering that area until the appropriate time. This tagging system provides built-in version control, ensuring that even with multiple shifts running, every section of the blueprint is copied exactly once. Once the work is done, the two new blueprints separate, and the cell completes its division right on time. We can view this entire process as a 3-step workflow breakdown. First, a specialized team physically unzips the blueprint to expose the data. Second, chemical workers act as start pins so builders can efficiently reconstruct the missing tracks. And third, chemical tags provide version control, preventing the system from duplicating work. This combination of overlapping shifts and strict tagging allows the cell to meet every deadline, every single time.